audio. Very good afternoon to all of you. We are waiting for the more number of participants to join this webinar. So we request all the participants, please be with us. We'll start this webinar after 10 minutes. Thank you. Once again, a very good afternoon to everyone. As we all know, India is celebrating second National Pharmacovigilance Week commencing from September 17 to 23, 2022. On the day 5 of this celebration, National Coordination Center, Pharmacovigilance Program of India, Indian Pharmacopoeia Commission is organizing a webinar on utilization of VigiFlow and VigiLyze, ensuring quality of ICSRs and signal detection. So I welcome all the members for this webinar. As we all are very well aware that NCC PVPI IPC is designated as a WHO collaborative center for pharmacovigilance in public health programs and regulatory services. Being a WHOCC, PVPI is regularly providing support to the WHO capacity building activities in CRO countries by conducting trainings, workshops, webinars, and also sharing the drug safety information such as drug safety alerts, PVPI newsletters through emails. The objective of this webinar is to strengthen the pharmacovigilance system in CRO countries. With this word, I would like to invite Dr. V. Kalai Salvan, Senior Principal Scientific Officer, Indian Pharmacopoeia Commission, for his remarks. Over to you, sir.
Good morning, good afternoon, and good evening to all of you. On behalf of Indian Pharmacopoeia Commission, WHO Collaborating Center for Pharmacovigilance in Public Health and Regulatory Services, I extend my warm welcome and greetings to all of you. So, on behalf of Indian Pharmacopoeia Commission, I extend my warm welcome to Dr. Adrian, WHO CRO, Dr. Noha from WHO Headquarters, Dr. Jennifer from UMC and Tommy from UMC, Dr. Meera Desai, and my dear senior colleague Dr. Jayprakash and all other experts, those who have joined this webinar. I also extend my warm welcome to all the delegates, those who are attending this webinar. We as a WHO collaborating center, we are extending our services to WHO member states, particularly in capacity building, training and other related areas of pharmacovigilance. The objective of this today's webinar is to update you about the developments and new versions of VigiFlow and VigiLyze so that the member states can effectively utilize these tools for identifying new signals and also for data management. So we take this opportunity to express our sincere thanks to WHO Headquarters, WHO CRO and DMC for extending their support. And I'm sure this webinar will be beneficial for all the member states in terms of using these tools for effective data management and signal detection. So without wasting much time, once again, on behalf of Indian Pharmacopoeia Commission, I extend my warm welcome to all the delegates. I wish a grand success of this webinar. Thank you, and over to Nikita.
Thank you so much, sir, for sharing your valuable insights with us. To address the gathering, I would like to invite Dr. Jay Prakash, Senior Principal Scientific Officer, Indian Pharmacopoeia Commission, for his remarks. Over to you, sir.
Thank you, Nikita. Good morning, good afternoon, and good evening to everyone. Respected all the international delegates, international delegates, and the experts of today's session, a very warm welcome for connecting with us, for joining the international webinar on the utilization of VigiFlow and VigiLyze, ensuring quality of ICSRs and signal detection. As you all know, that the quality, safety, and efficacy of medicines is of paramount importance, and India is one of the lead producers of the medicines and exporting them out to many countries of the world. Management of the safety data of medicines is a Herculean task, and we are grateful to WHO, Geneva, Switzerland, as well as the WHO UMC Sweden for providing us constant support for the use of the VigiFlow. VigiFlow is a web-based ICSRs management system, and many national centers, including India as on date, are sending the safety data of medicines through this gateway, which becomes available to our country as and when it is required. And we also have the access to use the VigiLyze, which is a very important analytical tool to see the status of the ICSRs from other countries of the world. And I am pleased that my colleague, Dr. V. Kalaiselvan, Dr. Sasibhushan, Mr. Vipin Sharma, Nikita, and all the colleagues of the Pharmacovigilance Program of India have done a wonderful job for organizing this international webinar. And we have the speakers who are really having in-depth knowledge in the area of pharmacovigilance. And today we have the registered delegate from 7 countries: Bhutan, Thailand, Myanmar, Zimbabwe, Yemen, Malaysia, Sweden, as well as from Switzerland. Each of the expert who has been identified for today's international webinar will justify their time and will throw the light in depth how to use the VigiFlow and how to use the analytical tool that is VigiLyze, and how this whole set of the safety data can be managed. As you all know, that India is one of the largest Asian continent, one of the largest country in the Asian continent, and has strength in many sectors, including pharmaceutical sector, IT sector, and many more sectors. And I will not be taking much time, but will be sending you my warm greetings to all of you, that this conference, this webinar will be one of the most useful and memorable event for you. If you come across any sort of the questions, either during the webinar or later on also, the team from the PVPI and the team from the Material Vigilance Program of India will assist you, and we look forward for your questions, for your queries, and the feedback in order to improve our future meetings. So with this, I again give my warm greetings to all of you and sparing your valuable time to attend this international webinar. Thank you very much.
Thank you so much, sir, for sharing your encouraging words with us. Now, I would like to invite Dr. Adrian Inobli, Regional Advisor for Medical Products Quality and Regulation, WHO CRO Office, New Delhi, for his remarks.
Okay, so good afternoon, colleagues, and thank you very much for inviting me in participating in this celebration of pharmacovigilance in India and also beyond in the region of Southeast Asia. So I'm first of all very pleased to see that the IPC has WHO Collaborating Center for Pharmacovigilance in Public Health Programs and Regulatory Services has organized this very helpful and also timely training in the context of the recent update of VigiFlow. As demonstrated during COVID-19, especially with, for just one example, but the thrombosis with thrombocytopenia syndrome, that TTS, the quality of the individual case safety reports is really essential to ensure the safety of the products that are on the market. So ICSR are one of the essential sources, and there are others, publications, studies, and so on, sources of information for signal detection, and the WHO, supported for a very long time by the UMC, have provided a set of tools to support member states. So there is VigiBase, which is a WHO global database for all the ICSRs, VigiFlow, which is the web-based management system for the ICSR. You're all familiar with this one, and VigiLyze, which provides an overview of VigiBase and which is also used to calculate, for example, disproportionality. And so the IPC has developed some expertise in also all these fields, and I'm very happy they can share this expertise with colleagues in India, but also beyond. So of course, all these tools do not replace a good and in-depth assessment of the cases at the national level. That's always very important. However, they can assist countries in completing their investigation and in triggering alerts and also complement their vigilance system. So it's still a very helpful and valuable tool. In general, VigiBase is really a common good which eventually benefit all member states through the detection and the assessment of signals, which are then shared in the WHO pharmaceutical newsletters. So whatever goes into that database really in the end benefit all. So today you will learn about the updated version of VigiFlow, standardized drug groupings, signal detection using VigiLyze, and so I wish you all a very productive session, and we're really looking forward to further answering your questions and discussing with you at the end of this session. Thank you very much.
For sharing your warm words with us. Now I request all the participants, please turn on your cameras for the group photograph. Thank you, everyone. Now, we'll start the technical session of today's webinar. To take the first session, I would like to invite Jennifer Wall, PV Officer, WHO Uppsala Monitoring Center, Sweden, on an overview of updated VigiFlow. We have received the recorded session of Jennifer. I request IT team to share.
you for inviting UMC to speak about updates in VigiFlow today for the second annual Pharmacovigilance Day. My name is Jennifer Wall, and I am a Pharmacovigilance Officer with Uppsala Monitoring Center, and today I will be covering an overview of updates in VigiFlow. But before I go into any of the updates in VigiFlow, it would be nice to go over a little bit of background about UMC and where we come from. The thalidomide disaster in the 1960s was the starting point of pharmacovigilance and medicine safety. At the time, there was no global system in place to collect information about adverse events. To prevent future tragedies of this kind, in 1968, the WHO set up a pilot project in Geneva in collaboration with a small group of 10 countries. This was later made permanent as the WHO Program for International Drug Monitoring, with the short name WHO-PIDM, and operational activities were moved to the WHO Collaborating Center for International Drug Monitoring, setting up in 1978 in Uppsala, under the sponsorship of the Swedish government. It was not until the mid-90s that the field name Uppsala Monitoring Center was adopted. In 2022, the 10 countries have become one. The original 10 countries have expanded to 153 full members and 22 associate members. The WHO PIDM covers approximately 99% of the world's population as of September 2022. Independent nonprofit foundation established in 1978 to coordinate a global collaboration for safer medicines. UMC oversees the technical and scientific operations of the WHO-PIDM with more than 170 members. We are the custodian and manager of VigiBase, whose database for suspected adverse reactions to medicine products used to generate insights for research and the medical industry. Developer of the WHO Drug Global, the world's most widely used drug dictionary. This is just a little bit about who UMC is. I'd like to just also offer up that UMC has education and training focusing on pharmacovigilance, both online and self-paced e-learning courses and face-to-face training courses that will cover many of the topics that I'm going to be going over today, as well as my colleagues later in the day. So today's learning objectives is to provide a practical training in the context of recent updates of VigiFlow, to build up your national capacity in pharmacovigilance in the 0 countries, and also to provide training in utilization of VigiFlow and VigiLyze while ensuring quality of ICSRs. VigiLyze and signal detection, my colleagues will continue to go into detail on presentations later in the day after this presentation on VigiFlow. What is VigiFlow? VigiFlow is a web-based ICSR data management system. It is your country's own national pharmacovigilance database, which supports the collection, processing, analyzing, and sharing of ADR and AFI reports. Some of you have seen this picture over time in many presentations, but for those of you who have not, this is Ed Napke with his pigeonhole approach to organizing ADRs back in 1965. So you don't have to manually have paper ADR forms and cubicles. VigiFlow provides you with a digitized ICSR data management system that sorts all this paper for you. And let's never forget, all these papers represent patients' lives. This is an overview of UMC's offerings to the WHO-PIDM members. VigiFlow is a central point as it is the National Safety Database. It also allows for direct and indirect data entry at 3 organizational levels: national, province, state, district. Different in-channels to VigiFlow are available here on the left-hand side. We have online reporting forms that allow configurations, an industry reporting module with full E2B compatibility, data entry via apps like VigiMobile, which is free for those with VigiFlow, and interoperability with electronic health records and other external systems via the ICH E2B standard. Today I will go into more detail about what's new with VigiMobile and industry e-reporting. After entering data in VigiFlow, the reports can be sent to the WHO global database called VigiBase. As part of assuring quality, someone usually at the NC level needs to actively approve each report before it's sent to VigiBase. My colleagues will cover what VigiLyze is here in the loop later this afternoon and go over standardized drug and reaction terminologies, WHO drug and MEDRA, that are built into all of these systems. Presently, we have 108 WHO PIDM members using VigiFlow. Most of you joining this session are users of VigiFlow, and maybe some have their own ICSR management system. So we'll just jump into some of the new features. Updates in VigiFlow include vaccines in VigiFlow, VigiMobile, AFI form in collaboration with WHO, AFI dashboard is an ongoing collaboration with WHO, the hierarchies available in VigiFlow on first, second, and third levels, industry e-reporting, and the new geographical information system via WHO. So, what's new with VigiFlow? I'll begin with the newest and most exciting, VigiMobile. You can always visit UMC's website after today's session to get more details on VigiMobile. What is VigiMobile? VigiMobile is an app specifically developed by UMC for field reporting of adverse events following immunization. Immunization workers can use it to collect data on their smartphone or other mobile device even when they are offline. VigiMobile uses the WHO standard AFI reporting form based on the 25 core variables recommended by WHO for collecting AFI data to ensure that qualitative data is available for analysis. Reports can be sent directly to VigiFlow as soon as there is an internet connection. When a vaccine is administered, the patient may experience an adverse event. Health workers organizing vaccinations in rural areas will manage that event, but they also need to report it according to National Vaccine Safety Surveillance Guidelines. Due to traditional work practices, limited internet access, or high internet costs, this process is often paper-based, leading to delays in information being sent to district or provincial level, where events or clusters of events may trigger more in-depth investigations. To shorten lag time and relieve workload on data entry staff, UMC has developed VigiMobile and VigiFlow for AFI data management and analysis system. Using their phone, tablet, or laptop, health workers can now capture events as soon as they happen, even when they are offline. This reduces the risk of vital information being lost in the field. If the app is online, the report will be transferred to VigiFlow within seconds. If offline, the report will be queued for sending later. It can be sent as soon as an internet connection is available. At national level, reports can then be shared with WHO's Global Safety Database, VigiBase. Report routing. A distributed workflow is supported in VigiFlow, allowing districts and provinces to manage their own data in the system. It is therefore possible to route reports from dedicated reporting areas to specified districts or provinces for data management and review. Once a report is received in VigiFlow, it is immediately available for review and further analysis at district level. At the same time, this report is also accessible to provincial and national program managers, increasing transparency and minimizing lag time in urgent situations. Only national level data managers can share information with WHO's Global Safety Database, VigiBase. If you'd like to try out VigiMobile, point your phone at the screen, scan the QR code with your camera, and fill out the form. I will leave it up here for about 5 seconds. You can also download it as an app to your computer, Android, iPhone. Scan the QR code and you can have it directly in your phone. And this is what it would look like on the interface in your phone. The interface will look like this on your computer, Android, or iPhone, where all the fields will be able to be filled in. We have several online reporting tools. Here's a comparison of all the features. What's important for VigiMobile is the possibility to fill out and store reports offline. And if regions or province offices in the country are responsible for handling the reports from within their region, VigiMobile can be set up so that it forwards the report to the right region level inside VigiFlow. VigiMobile uses geographical information provided by WHO GIS, which is better than free text geographical information when you use it for statistics and other purposes. In response to the unfolding monkeypox situation, we have added relevant vaccines to the short list of vaccines that users will find in the app. And currently we have 6 available languages pre-configured. The report flow with VigiMobile for APHIS. The vaccine is administered. The patient experiences an adverse event. Health workers manage the adverse event and report the 25 core variables with VigiMobile. If the health worker has an internet connection, the report will be transferred to VigiFlow within seconds. If they are offline, the report will be queued for later transmission. The National PV Center will be able to see the APHIS-specific information in the extended APHIS view, which also displays all the 25 core variables, even those that can normally not be captured with the E2B standard. An APHIS line listing can be generated for analysis or for sharing with relevant stakeholders like the National Immunization Program or safety committees, but it is also possible to provide the immunization program direct access to VigiFlow, and possible to route reports that are sent via VigiMobile directly to a sub-organization, for example, a province or state level center. From the National PV Center, the data can be shared with the WHO Global Safety Database, VigiBase, to be analyzed and VigiLyze in a national or multinational. Under the pandemic, the APHIS tab for 25 core variables was developed in alignment with WHO. With VigiFlow, focus has been on improving usability in the vaccine surveillance area. We have basically digitalized the WHO APHIS reporting form as one major factor for long lag times, is that the paper forms takes a long time before they end up being entered into the system. There was a high threshold for data entry staff to use the existing ICSR view to enter the information. This work was done in tight collaboration with WHO. We also ensured that all essential information can be analyzed using an APHIS-specific Excel export. further improvements are underway. AFI dashboard is coming soon. It's a UMC close collaboration with WHO to bring EPIs to this platform. The development is ongoing. What it would look like. So capturing geographic data on patients, reporters, and health facilities in a structured format for statistical follow-up helps identify reporting clusters. VigiMobile uses health boundary data provided by the WHO GIS Center for Health. National immunization programs in different countries are often quite similar in structure, depending on the population size and area covered. The setup is often similar to the one shown here for the imaginary country of Harmonia, which is divided into a number of provinces where each province has several districts. Each district may have a number of sub-districts or public health centers, clinics, etc. The WHO GIS data in VigiMobile typically supports collection of structured geographic data up to district level. VigiFlow for AFI dashboard. The training environment would support e-learning VigiFlow for AFI to be released later this year. It will provide statistical views for descriptive epidemiology and surveillance performance. Now I'm going to cover hierarchies in VigiFlow, the 3 levels. One of the new updates in VigiFlow is that the National Center can see everything in real time. If entered at a lower level, the NC will see it immediately. There's also some support for workflow, assigning reports to users and organizations within your setting. Fun fact, Philippines has over 2,000 regional centers on these level 2 and 3. India uses level 1 for their 250 AMCs. So whichever level and workflow works best for your country is what you should be using. What it looks like in VigiFlow. Under the VigiFlow Manage Accounts tab, you'll be able to open up to 3 levels of organizations. And this allows you on the NC level 1 to be able to add and delete users yourself without having to contact UMC, but contacting your national center instead. Another new exciting add-on feature in VigiFlow I will cover is industry e-reporting. Purpose of industry e-reporting: to further the capacity to share information and to enable ICH compliance between marketing authorization holders and NRAs. What exactly is it? It's a one-way submission tool for non-E2B and E2B industry to be able to submit to the country's national regulatory authority. This relieves the burden on the national center to be entering all the data for the marketing authorization holders and puts the responsibility on them. If you are in doubt about something new in VigiFlow that just automatically is updated, please feel free to always check UMC's website, where we regularly upload release notes detailing what we have changed in the system. How you use your system for quality of ICSRs depends on each person in the overall process. There are many steps to enhance your data quality process. Plan for good quality data. So ahead of time, have SOPs for your facility, training internally and externally. Always feel free to contact UMC for any PV training guidelines that we have. To enhance the data quality in VigiFlow, check for duplicate detection. Before you enter your ICSR, check to make sure that the ICSR is not in the system by using the filter function in VigiFlow. Assess your ICSRs for quality and completion. If there are missing fields and data, contact your reporter and see if you can fill in the information that's missing beforehand. Of course, in reality, reports can often lack some of the essential information you would need to be able to clearly assess whether the drug likely caused the reported event. In reality, we need to face and deal with the limitations of a spontaneous reporting system, which is faced with many challenges. In this instance, it seems as if the one drug is what caused the adverse drug reaction, but in reality, sometimes it's concomitant drugs that the patient is taking. So what information is essential information for assessment of a case? In an ideal world, what information would we like to have on all reports? You would like to clearly understand who the patient is, underlying diseases, etc. You'd also like all possible information about the reaction, what happened and when. The chronologic events, was it serious, was it severe, was medical or other treatments required? Did the patient recover? About the drugs, you want to know exactly what product it was, the ingredients, formulation, route of administration. Also, of course, the dosage taken, dates of administration, and if the drug treatment was stopped. And there are a number of other items that would complete the picture of the case, like any lab tests, clinical examinations, etc. What you put into your national database, VigiFlow, is what you get out of VigiBase for quality and signal detection. Whether you are entering data at the subnational level or quality control at the national level, each person's contribution to the ingredients plays a major role for significant signal detection. Thank you so much, and we will be available from UMC later live for the questions and answer session.
Thank you, Jennifer. Jennifer is also present with us. I request all the members, if they have an inquiry, they can directly ask.
Okay. So if I may, maybe I have just one question. So we saw the new VigiMobile application, so that's indeed something very interesting. I was wondering, so for the time being, it's only on AFIs. So I was wondering whether there was any project in the future to extend it to adverse drug reactions, and if so, which maybe would be the timelines. I think that could be helpful for colleagues connected here, and especially considering also the particularities in the regions. So you mentioned a couple of languages, but except English, none of the languages are much talked in the region. So do you feel that it may be easy for countries if they wanted to expand, for example, the language of the app to other regional languages? Thank you.
Thank you for your question, Dr. Nobly. Yes, we do have adverse drug reactions in the pipeline. I can't say for sure what the date would be, but that is a request that we're considering in the future. Presently, you can put, if needed, in the free text field in the app, but it has not been coded to ADRs yet. And when it comes to languages, yeah, we do have those 6 languages available now, but as far as in the future, there is consideration to always add languages as they come in and what the needs of the countries are. So we would take that into consideration in the future for different languages that aren't available at the present.
I am Dr. Jayaprakash. It was an interesting presentation and I have a comment. It is a very good application for mobile use. I just want to know whether it is downloadable on charge basis or free of charge.
It is free with all VigiFlow using countries. So if you already are a member of the program and you're using VigiFlow, you just need to contact us, and it's a free add-on.
Okay, very good. Congratulations, Dr. Jennifer.
Thank you, Dr. Prakash.
Thank you, Dr. Jennifer, for the brief explanation of VigiFlow updates, global AEFI reporting, VigiMobile, report flow in VigiMobile, quality of ICSR. Thank you.
Thank you very much.
For the next session, I would like to invite Tommy Drews, PV Education Specialist, WHO Uppsala Monitoring Center, Sweden, as on WHO drug standardized drug grouping by VigiLyze. We have also received recorded session of Tommy Drews. I request IT team to please share.
Hello, my name is Tommy Zeus, and I will give this presentation on WHO drug standardized drug groupings in VigiLyze. I will quickly introduce myself. I am a pharmacist and chemist, and I work as a pharmacovigilance education specialist at Uppsala Monitoring Center. Previously, I have worked with maintenance of the WHO Drug Global Dictionary and been involved in the maintenance and development of standardized drug groupings. Here is today's outline of the presentation. First, we'll go through the objective, then an introduction to VigiLyze, an introduction to standardized drug groupings, SDGs, as I will call them from now on, the SDG user guide, how SDGs may be used for analysis, and a conclusion. The main objective that we will focus on in this presentation is to provide training on WHO drug standardized drug groupings using VigiLyze. Before we dwell into the standardized drug groupings, let me say a few words about VigiLyze. VigiLyze is a signal detection and signal management tool provided by UMC. It supports national signal management processes, including qualitative assessments. It uses insights into the safer use of medicines from members of the WHO Programme for International Drug Monitoring as a starting point for efficient quantitative signal detection. VigiLyze provides a global context for your national data through its close integration with VigiBase, WHO's global database of reported potential side effects of medicinal products. VigiLyze is provided free of charge to national pharmacovigilance centers in all members of the WHO Programme for International Drug Monitoring, where users have their own login details. Major strengths include the ability to calculate disproportionality based on any chosen country or group of countries within seconds, and then recalculate when new filter criteria are chosen. It has ready access to related investigations shared nationally or globally when searching for the same or similar medicinal products and reactions. By sharing their data and knowledge with the global database, member countries help increase each other's understanding of potential safety concerns and then reduce the risk of harm from medicines. Access to VigiLyze is found on the webpage vigilize.who-umc.org, and in this presentation, some familiarity of VigiLyze can be useful. Right, let's move on to SDGs, the standardized drug groupings. How do you find them in VigiLyze? In the qualitative view, you can find all the SDGs through the filter by drug and by clicking the SDG view. Then a list of main SDGs should appear, where you can filter by main SDG or by going into subgroups with the arrow and narrow down the search to a specific subgroup or a selection of drugs within an SDG or several SDGs. Instead of using the filter, if you know exactly which SDG or subgroup is of interest to you, you can also search for it directly in the search box, like you would a drug, reaction, or country. So here I searched anticonvulsant, and it suggests the main SDG anticonvulsants or the subgroup other anticonvulsants. Those of you familiar with VigiLyze already knew this, so let me move on to a topic of interest. What are SDGs? The definition of a standardized drug grouping is any grouping of medicines having one or several properties in common. The individual grouping can be based on indication, chemical properties, pharmacodynamic properties, and/or pharmacokinetic properties, as well as any other property of interest. So what is actually included in the SDGs? SDGs are based on preferred names for active ingredients in WHO Drug Global. So if we take a look at an example, if a substance, an active ingredient, meets the inclusion criteria and is added to the SDG, like enalapril in the angiotensin converting enzyme inhibitors SDG, then all relevant variants of that substance are added to the SDG as well. Then all multi-ingredients where at least one of the ingredients meet the inclusion criteria are then added automatically to that SDG or subgroup as well, even if the other ingredients are not part of the SDG or part of any other SDG either. An active ingredient or combination may be present in many different SDGs if they meet the inclusion criteria of those SDGs. Another important aspect is to understand the SDG structure. The different groupings vary depending on which SDG it is. First, we have the main or the parent SDGs. A few of them contain substances directly in this main SDG, whereas others may have subgroups within them. The subgroups can also have substances directly in them or further subgroups. These subgroups will then have the substances. So in an SDG, we can have the main or parent SDG, a subgroup level, another subgroup level, and then the substances. So we can take at some real examples. Let's start with the SDG benzodiazepines. In this SDG, we don't have any subgroups, so the substances are in the main SDG. There are also other examples for SDGs without subgroups, including antiemetics and antineurgens, corticosteroids, statins, and the World Health Organization model list of essential medicines. Most other SDGs, however, are built up with subgroups. And here is an excerpt from the cancer therapies SDG as an example, where several subgroups exist. And in the actual SDG, there are many more subgroups. This is just to illustrate that subgroup levels may exist within SDGs. First, we have subgroups, which either have subgroups within them or substances in them directly. Like the targeted anti-cancer drugs will have further subgroups, whereas the other antineoplastic drugs has no need for another subgroup, so the substances are directly in them. And if there is a subgroup, it will have the substances or the active ingredients in the second level subgroup. So this is how the SDGs may look. And to learn and read more about the SDGs, I recommend browsing through the SDG user guide, which can be found on the UMC homepage or directly in VigiLyze, if you have access. In VigiLyze, you will find the user guide by going to the SDG view, and here if you click on the information circle highlighted in yellow at the top right of this window, the user guide will open in a new tab of your browser. The user guide is updated with each release of WHO Drug, just like the WHO Drug Dictionary, every March and September each year, meaning that the SDG content may also update in March and September. The user guide gives a deeper introduction with the background, definition, SDG hierarchies, and further details. It also provides information on each standardized drug grouping. For each SDG, you will find a short description of it, the inclusion and exclusion criteria for drugs. So if the drug qualifies to be in the SDG. Then there are details on the subgroups within the SDG if there are any. There is also scope information and other recommendations or points to consider with that particular SDG. Then you have the scope, for all SDGs. All drugs are assigned a scope, whether it's narrow or broad. Narrow is the default scope in most SDGs. And the scope can have a variety of different uses, and it is stated in the definition of the individual SDGs in the user guide. The scope can indicate clinical relevance, the main indication, or what kind of substance. It may be like a herbal substance. This table shows a few examples of current standardized drug groupings in different categories. It shows 4 main SDGs categorized by a certain property, like anti-infectives, which is indication-based. Then we have chemical properties like benzodiazepines, or pharmacodynamic properties or mechanism of action, like the drugs acting on gonadotropin-releasing hormone receptors, and pharmacokinetic properties, where drugs interact with a certain enzyme or a transporter protein. And these are only a few examples of SDGs. There are currently 47 main SDG groups, and plenty have subgroups within them. And a lot of time can be spent on going through each SDG. Instead, I will show you an example with a drug. Here we have then an example to show when a drug has been selected in VigiLyze in the SDG view. The box is marked in which SDG the drug is present with a line. In the picture, they are also highlighted in yellow for clarity. This shows that the drug has properties that meet the inclusion criteria to fit in several SDGs, as the drug possesses many properties based on indication and pharmacokinetic properties. All right. So this has been a quick overview of what SDGs are and how they are found in VigiLyze. So you may ask, how can they be used? It depends completely on the drugs or properties of interest, and there is a lot of potential. In the pharmaceutical industry, SDGs have mainly been used as drug lists of prohibited medications in clinical trials. Here I will name a few other possible uses for analysis. So one SDG contains drugs for COVID-19, and the subgroup is the vaccines, and further divided into the different types of COVID-19 vaccines based on the vaccine platform. This means that the user can easily find all cases of reports from one or several platforms of COVID-19 vaccines. The user can choose the entirety of the subgroup, or narrow down the search, or broaden the search as they deem necessary. Another possibility is to broaden a search based on drug properties. For example, a particular reaction with a group of drugs instead of a single ingredient in that group of drugs. And in this example, I have taken the SDG corticosteroids and the preferred term dyspnea as a reported reaction. As you can see, this has broadened the search vastly compared to only having one corticosteroid in your search. and patterns may emerge in your search. So this type of query may enable the user to find more cases fitting a certain profile. And if needed, the user can narrow down the search using the filter to fit your search criteria better by, for example, removing some of those corticosteroids or adding additional drugs that are of interest, and likewise with the reactions. Another possibility is to look at drug properties from a different point of view, such as pharmacokinetic parameters or mechanism of action that may affect a drug safety profile. Examples include using the SDGs where drugs interact with CYP enzymes, UGT, or PGP as inducers, inhibitors, or substrates, or looking at the NMDA receptor agonists and antagonists. In this image, to demonstrate the functionality, I have filtered the cases to contain all NMDA antagonists that are also PGP substrates, and where the reported reactions are acute kidney injury, as well as patent ductus arteriosus. There are many possible combinations to search and use SDGs depending on what drug property is of interest when performing your analysis. This is an example where standardized drug groupings have been used in signal management and signal assessment. This is a UMC poster from 2019 to illustrate that the utilization of the SDG anti-angiogenic drugs with antagonistic effect on the vascular endothelial growth factor receptor, inhibiting actions on vascular endothelial growth factor or its receptor tyrosine kinases, helped refine a signal between a drug nintedanib and a reaction colitis. Nintedanib, the drug, is a small molecule tyrosine kinase inhibitor, and it inhibits the platelet-derived growth factor receptors alpha and beta. It inhibits fibroblast growth factor receptors 1, 2, and 3, as well as the vascular endothelial growth factor receptors 1, 2, and 3. And due to this mechanism of action of the drug nintedanib, patients might have an increased risk of gastrointestinal perforations, and so ischemic colitis, according to the Summary of Product Characteristics, the SPC. The conclusions drawn in this screening were that standardized drug groupings are a useful tool in the signal management process. Refinement of the signal nintedanib colitis using an SDG provided a stronger evidence of a potential causal relationship compared to ATC. Exploration of potential drug class effects during signal assessment may be improved by use of the WHO drug SDG. So, to the conclusion of this presentation. An SDG is any grouping of medicines having one or several properties in common. SDGs can be used in different ways depending on what drug property is of interest. And the groupings in the SDGs can aid in signal assessment. And with that said, I welcome any questions or suggestions that you may have. They may be on standardized drug groupings, VigiLyze, any functionalities, how you search. Any questions or suggestions are welcome. And as always, you can contact us via email to vigibase@who-umc.org if you want to ask any questions or have any suggestions in the future.
Thank you. Now I request all the members, if they have any query regarding the session, they can ask. Tommy Drews is present with us. You can ask.
Okay, so one question on my side. Thank you very much for your presentation. I was wondering with respect to the SDG, maybe I missed it from your presentation, but I think sometimes maybe there is some choices that need to be made. So I was wondering how you were constituting this SDG and deciding which drugs goes where and how it is related. And especially, I think you mentioned indications, but often indications are not necessarily harmonized worldwide. So I was wondering a little bit how you made these calls, just to know how this in the future may also impact on the assessment and the way the tool is used. Thank you very much.
Thank you very much for your question. So when creating the content in the standardized drug groupings, we use relevant literature and summary of product characteristics depending on what property it is. Let's say we have a drug that is considered as an anti-infective, an antibacterial, then it has to be stated clearly in literature or summary of product characteristics that it has been used at least in one country in the world. So it may be used as an anti-infective, antibacterial in one country, but perhaps not another. And the structure of the groupings are to be quite generous. Since this is for global use, if it is considered an anti-infective in one country, it may not be relevant in another, and so it is important to filter what is relevant to you. Thanks again. I hope I answered your question. And
Thank you for the yes. Go
I
ahead.
can also add that some SDGs are based on the ATC classification. So that is also one reference that we use because we know that ATCs are based on use of drugs. So if an ATC is given to a particular drug, then we consider that as a relevant reference as well. Thank you.
Thank you, Tommy Drews, for the nice presentation and the detailed description of VigiLyze, SDG structure and scope and user guidelines. Thank you. Now, it's time for the last session of today's webinar. For that, I would like to invite Dr. Meera Desai, member of Signal Review Panel, Pharmacovigilance Program of India, on signal detection by using VigiLyze. Over to you, ma'am.
Can you see the slides?
Yes, ma'am. Slides are visible, ma'am.
Okay. Thank you, Nikita, and Dr. Kalai Selvan, Dr. Jai Prakash, Vipin, and the Signal team at the National Coordinating Center, PVPI, all the international participants, the participants from the Regional ADR Monitoring Center of India. Very good afternoon, good evening from wherever you are. It's literally a pleasure for me to be a part of this webinar. And thanks PVPI for this opportunity. It's always a pleasure to be associated with the PVPI for any matter related to the adverse drug reaction monitoring. So for the almost next 1 hour, we'll be mainly talking about the signal detection using VigiLyze. And the outline of my presentation includes what is a signal, what is the whole plan and the process, what are the qualitative and quantitative methods for signal detection, what is the whole process of signal detection at PVPI, and the outcome and the impact of PVPI as far as the signal detection is concerned. So if we look at this whole process of signal detection, it's highly complex, technical, and it's like hunting a needle in a heap of grass. And if we look at the role and responsibility of pharmacovigilance professionals, it's like detectives, you have to undertake the whole investigation process, coming across any adverse reaction associated with drug. And the pharmacovigilance professionals are the backbone of this whole investigation process. Now, the big question for the pharmacovigilance professional is there any causal relationship between the drug and the adverse event? Whether it is unknown or less commonly known risk? Is it by chance or is it significant? Is it significant enough to raise an alarm? Is it relevant for the healthcare professionals for a wise therapeutic decision? And is it out of proportion than expected? So these are the big questions which the pharmacovigilance professionals, they have to undertake. They are faced while doing this whole exercise. So with this background, let's talk about the definition of signal as WHO defines. It's a reported information on a possible causal relationship between a drug and adverse event, the relationship being unknown or incompletely documented previously. While the COMS, the definition is the information which arises from one or multiple sources, which suggest a causal association between a drug and an event. It may be a beneficial or an adverse association, and it has to be judged to be of sufficient likelihood to justify verificatory action. In short, both the definitions, the causal association between the adverse event and the drug, needs to be verified. It has to undergo the whole investigation process. Now, what is the whole plan and the process which is undertaken at PVPI as well as the UMC? The first and foremost thing is the VG base, one of the largest pharmacovigilance database of the adverse reactions associated with the medical products by WHO. It is one of its kind in the world. And that is also all the adverse reactions which are submitted to the National Coordinating Center into the VG base that is initially screened for unlisted drug adverse events. Now, what is actually identified? The adverse reactions, which are serious, which are associated with the new drugs, which are found in some spatial population, which are likely to cause some risk to the public health at large. they are identified. So this is the first step which is to be done. And once these unlisted drug adverse event reports are identified, they are segregated for initial manual assessment to generate a hypothesis. For example, a drug XYZ can cause this reaction. And at the same time, the drug and the adverse reactions are also quantitatively analyzed by giving the disproportional analysis. We will be dealing with that in detail. And once this ICSR causing certain reaction is identified, they are submitted for in-depth analysis, especially the clinical assessment, the quantitative assessment by a multidisciplinary team known as a signal review panel. And this is submitted quite in advance, especially let me share with you of our National Coordinating Center. They share with us well in advance because there is a lot of homework which is to be done by the panel members, and a fixed time is decided. And at the time of this meeting, each of these drug adverse event listed, identified, they are discussed in detail. There is literally presentation. There are deliberations. The subject experts are also invited. And at the end, the recommendations, the subsequent action is decided. Now, from this slide, it looks very simple and easy. However, it is not that. It is highly complex. It requires a lot of homework, literature search, in-depth analysis by all the stakeholders before you come to a conclusion. Now, what are the sources of signal detection? As far as our pharmacovigilance program is concerned, the national and the international program, we are looking for the spontaneous report which are submitted by all the ADR monitoring centers to the national database. So these are the spontaneous report. However, there are also sources like published case reports, case series, post-marketing surveillance reports, clinical trial data, pharmacoepidemiological studies, various other regulatory database of USA and UK, registries maintained by the government or even the professional organization can also become a source of signal detection. Now, what are the different methods of signal detection? Broadly, there are two. One is a traditional or a qualitative assessment, and the second is the quantitative or applying the statistical tool. The qualitative assessment, which is done on case-to-case basis, it is actually a manual assessment of the individual case safety reports, case series, the PSUR, post-marketing surveillance report, or the periodic safety update reports, or other even sources. So the qualitative assessment, even the Bradford Hill criteria can be used. However, if you look at the database, it is not suitable for the large database analysis. And our VG base, there is an exponential growth, the volume of ICSR, which are increasing. And this I couldn't get it from the WHO website, UMC website, that if you stack these ICSR one above the other, they will even cross the height of Burj Khalifa. It is running in millions today. And this manual assessment of these ICSR in the VG base is practically not possible. And that is why we need to switch over to some automated system that is applying the statistical method so that we can quantify and identify for the subsequent action. So the application of the statistical methods to the database, pharmacovigilance database, that is known as a data mining process. It's a process of applying statistical algorithm to your database, which generates a statistical value or a score. So in short, it quantifies. And that quantitative number, the score, that indicates the strength of association and the potential safety issue. Further, higher the score, stronger is the statistical association, and it definitely complements the qualitative approach. And the most common method which is used is a disproportionality analysis. That is the backbone or the driving force which is used in our database. The fundamental principle of the disproportionate and proportionality is to detect the unexpectedly frequently reported drug adverse event as compared to the relative to the background of other reports. And this helps to quantify the degree to which a drug and an adverse event combination co-occurs disproportionately compared to what would be expected if there was no association. In short, this is a tool to generate hypothesis on a possible causal association between a drug and an adverse event, which is to be followed by clinical assessment of ICSR. In short, if I try to simplify the disproportionality analysis, what we observe is different than what is expected. That is disproportionality. So what are the quantitative parameters which are used in this disproportionate analysis? The first and foremost is a proportional reporting ratio. This is very commonly used because it is easy to interpret, and it fundamentally, this mainly the value which is generated by this PRR assessment, it indicates how many times higher is the risk for an adverse event, especially in a population which is exposed to the drug compared to the population which is not exposed. That is a fundamental principle. And there is a formula, and the PRR, which is either 2 or more than 2, it is considered that yes, there is definitely some importance or it is significant. The second quantitative parameter which is used is a reporting odds ratio known as ROR. And again, there is a formula. And both of these PRR and ROR can be derived using 2x2 contingency table. Further, the third parameter is an information component, which is based on the observed and expected number of case reports. If the value is positive, which means anything more than 0, that means higher reporting than expected. Moreover, chi-square TER is also used, whether there is a significant difference between the expected and the observed frequency, which should be either 4 or more than 4. And last but not the least, that is number of drug event combination. And the number, the magic number, which is given obviously because of certain reason, it should be either 3 or more than 3. And this is based on the incidence spatially. Now, these last 2 slides looks very complex, and it's more of a job of a biostatistician, which the medical persons are not much familiar. However, don't get worried. We don't have to calculate it individually. Let me share with you the fundamental principle that how this PRR is calculated. And this is what is showing. The slide is showing that this is known as a contingency table. What we need to identify is the reaction of interest. What I mean by reaction of interest, that if a drug adverse event combination is identified, the adverse event, that is, we are interested in that adverse event, and the D, that is a suspected causal drug. So what are the number of reports which we are interested in? They are in your database, which is caused by your causal drug, suspected drug. So that is A. And in the database, the same causal drug will also be causing some other reaction which we are not interested in, and that is B. Moreover, the reaction which we are interested, reaction of interest, are there any other drugs in the database which are responsible for the same reaction, all other drugs, which are also causing a reaction of interest, that is C. And lastly, all other reactions caused by all other drugs, that becomes your D. So this is the fundamental 2x2 contingency table. Now, let me give you an example. This is what we have already done at our signal review panel along with NCC, National Coordinating Center. In the database, the drug cefixime, let's suppose, or let's say that this is a real case, had a total of 5,189 adverse event reports, of which 79 were due to acute generalized exanthemus pustulosis, known as AGEP. This is a severe skin reaction, drug-induced. There are a good number of drugs responsible for the severe reaction, and it is a drug-induced problem, drug-induced adverse reaction. So in the database, these are the number of reports by cefixime, and out of this 79 are, this is the reaction of interest, that is acute generalized exanthemus pustulosis. The entire database has somewhere around 1,54,620 adverse report, that is our database, of which 175 is the total acute generalized exanthemus pustulosis, which includes caused by cefixime and other drugs in our database. So this is the data which is provided. Now, applying 2x2 contingency table, this is the 79 caused the reaction caused by cefixime, and the total number of reaction caused by cefixime minus 79, this is the total number of reaction, this comes to be B. And in the database, there were 175, our reaction of interest, AGEP. So that is minus 175 minus 79 comes to 96, that is C. And all other drugs in the database causing all other reports, this is from the total database. You apply, this is the formula which is already given, and by the calculation, the final figure comes is PRR value is your 25. Now, what does this 25 means? It indicates that the probability of reporting acute generalized exanthemus pustulosis rather than any other event due to cefixime is 25 times higher compared to the probability for the reference drugs. Reference drugs means the all other drugs which are also likely to cause the same reaction, like calcium channel blockers can cause, then beta-lactam antibiotics can cause, then macrolides can also cause. So there are so many other drugs which can cause this reaction. So it is compared with the drugs which are likely to cause, it is 25 times higher with cefixime. This is the analysis which is done. So by applying this formula PRR, what we have done is quantification of the reports which are in the database. Now, as I said, these are very complex calculation. The medical persons, we are not much familiar, at least I am not much familiar because I am not a statistician. So as UMC, Dr. Jennifer and Tommy has already, my previous speakers have said, they are coming, they have, UMC has come out with the excellent softwares, and that makes our job very easy. And this is done automatically, it is calculated by VigiLyze. And this is how it looks in VigiLyze. You can see that there are various other things which you have, my previous speaker has already very nicely, clearly mentioned, explained in detail. the overview, what is the database from India, what is the global database, what is the difference in the age, gender, etc., etc. What we are talking now is a disproportionality analysis, quantification of the number of reports caused by cefixime, and our reaction of interest is acute generalized exanthemus pustulosis. And as you can see that these all calculation is already seen, and it is calculated by this excellent software VigiLyze, that is IC values, PRR values, ROR values that is already calculated. You just have to identify what you want to search. Now, there is something known as a threshold value, and this is pre-decided. The threshold value for signal detection, at least I can say for our PVPI in India, the decision rule, the first and foremost thing is the PRR value has to be either 2 or more than 2. The IC value has to be positive. The chi-square test has to be either 4 or more than 4. And there has to be 3 or more cases to highlight the potential signal. In PVPI, any of the 2 above criteria fulfills a signal. And that we usually consider in the signal review panel discussion. So with this, let's try to find out what really happens at the National Coordinating Center, how the signal detection process takes place at PVPI. All of you know that currently there are more than 500 ADR monitoring center doing excellent job, and they submit the report through VigiFlow, gets into our database, and over a period of time, over the years, the number of reports, there is a substantial growth in our database, and India stands 9th as compared to the other country in reporting all the ADR, ICSR in the VG base. And there is still a scope of further improvement. Now, at PVPI, we have a signal review panel that is multidisciplinary, including the subject experts. And the first and foremost thing is to identify, as I explained you in my previous slides, the first is you have to find out, identify the drug adverse event combinations, which are unlisted, which are very rare, which are serious, which have a significant impact. So for that, to identify the potential signal, we have a standard operating procedure. This is documented, that which cases will be selected by the National Coordinating Center, which will be submitted for subsequent review. So this SOP is followed. There is an initial assessment of these ICSR for these unlisted drug ADR combinations, whether they are serious, not previously reported, ICSR, as I said, of special interest, adverse event not listed in the company's package insert or the product information leaflet. And then subsequently, the PRR value and IC value, the statistical values are also identified, assessed, and submitted. And this whole package, which are the drug and the adverse event combination, the number of reports, the IC and PRR values, they are then submitted to the signal review panel. And the agenda is prepared. What cases will be discussed in the next meeting? Let me tell you that we almost have 3 meetings, 3 to 4 meetings every year, every 3 months. So this is again a very big job, responsibility on the National Coordinating Center to do the homework, submit it to the SRP members. And the SRP members, they literally do again in-depth analysis, review of these drug adverse event combination. So these are the individual case safety report which are submitted to all SRP members, and that gives that how many are the number of cases in detail. Each of these ICSR of that drug adverse event combination, they are submitted to us. Now, we also have a template that how these cases will be presented. Each of these drug adverse event combination cases will be presented at the SRP meeting. This is a part of our SOP, standard operating procedure. What are the number of cases selected from PVPI database? The number of cases in the global database? How many of these? Now, this is the homework which we have to do as an SRP member. How many of these cases, the causality assessment, which is reported by the ADR monitoring center, they are certain, probable, possible, not mentioned. What is the time relationship? What is the action taken? The outcome of adverse reaction, the confounding factors, whether it is mentioned in the package insert, literature search, literally it is done, whether there are any case reports, whether it is mentioned in the textbook, reference book, if it is published in any journal, whether it is a PubMed index journal, whether the journal is peer reviewed, and which is the country or the authors, what is the location. We'll literally look, even meta-analysis. We'll literally do extensive literature search before the SRP meeting is, all members we get. What are the IC and PRR value and any other criteria which is that comes up during the discussion of SRP meeting. And this is almost I have described that what are the number of, this is the detailed assessment is done. Each of these, see, the SRP members not only do the homework, but at the time of SRP meeting, the signal review panel meeting, each of these ICSR, they are literally assessed, discussed, analyzed on case-to-case basis. And I think this is an excellent job, which is done by the National Coordinating Center under the leadership of Dr. Raghuveer Singh, Dr. Jai Prakash, and Dr. Kalai Selvan. And this is actually, this means whatever is decided, it is not out of blue. We literally have some strong evidence that is that is considered before coming to the recommendation and action. Even the patient outcome, how many of them recovered, how many did not recover, in how many cases it is the process of recovering. So whatever is mentioned in the ICSR, that is considered. What are the confounding factors? Whether the patient is taking any other drug, which also can cause the reaction. Is the patient is suffering from any comorbidities or the underlying disease process can cause? Causality assessment. What is the result of de-challenge, re-challenge? And this thing is almost discussed in detail, and the homework is done by the SRP members. And then at the time of SRP committee, the meeting, all these are discussed in detail, analyzed case by case basis, and the decision is made as per the clinical review, expert opinion, as well as statistical value. The recommendations are done. Now, let me share with you some of the real examples which we have done very recently. So this was the case which was discussed in this year, early this year. And I had this because I was a part of this discussion. So the drug adverse event combination which was identified, that was cephalosporin as a group causing fixed drug eruptions. And there were 186 cases, ICSR, in our Indian database starting from 2011 to January 2022. Look at this number of ICSR. The extent of homework which is required, we literally have to read this, go in detail. And when I was going through this, it was a marathon task. And how many of these out of 186 ICSR, they were related to the third generation cephalosporins. Cefixime alone, where only one drug was given, there were 78 cases. Other cases, there were other drugs. There were confounding factors like ornidazole. that is imidazole derivative, cefixime with ofloxacin, that is a fluoroquinolone. Then other third generation cephalosporins were also there in ICSR, ceftriaxone, ceftazidime, cefpodoxime, cefotaxime. So among this, the third generation cephalosporin, there were maximum number of cases. Moreover, there were also 7 cases from second generation cephalosporin. There were also 20 cases from first generation cephalosporin. And there were 10 ICSR reports which were not considered for assessment because the essential data was missing. Now, this is very important point for all the ADR monitoring center, that whatever is submitted, it has to be complete, the critical information, the complete clinical information which is required. If it is not fulfilled, that ICSR is useless. And this is a big job of all ADR monitoring centers. Moving ahead, from 186 fixed drug combination due to cephalosporin, there were 523 cases were reported globally. The causality assessment, that was also how many number of these in the bracket reports, that how many of them, majority were probable, followed by possible, and there were 16 cases which was certain. The time relationship was between 30 minutes to 6 days. 170 cases, there was positive D-challenge. There were 2 cases was positive re-challenge. Majority were recovered, followed by recovering. And obviously, there were confounding factors like concomitant medications, which are also known to cause. And interestingly, this adverse event is not mentioned in any of the package insert of USA, UK, as well as India. Moreover, an extensive literature search, we found that it's not mentioned in textbook, reference book, or meta-analysis, but there were case reports which were published in PubMed Index Journal. There was one induced by ceftriaxone in American Journal of Clinical Dermatology way back in 2008. Cefotaxime induced generous bullous fixed drug eruption in toxicology, that was very recent, 2018. And cefaclor induced fixed drug eruption in Annals of Dermatology. So even the subject expert dermatologists who are treating, who are seeing these patients, they have observed, they have come across this reaction, and there are published literature for that. Moreover, a big job was done by NCC, the signal committee members, that for each of these cases, IC values and the PRR values were even calculated. So this particular case, a lot of discussion took place, and the SRP committee recommended to include fixed drug eruption as an adverse drug reaction in the package insert, the product information leaflet of cephalosporins which are marketed in India. So you can see the impact, and this has been forwarded to the regulatory authorities. Now, let me share with you one more case. That is the, Montelukast induced tinnitus. Okay, yeah. The second case I would like to, bring to just for the discussion purpose. The second case which we came across was Montelukast causing tinnitus. This was last year, we discussed it. There were 10 cases in the VG PVPI database, and the number of cases globally was 73. There was one case which was certain, followed by possible and probable. The time relationship in majority of the cases was 8 days. The positive D challenge was observed, which was reported in ICSR, was 5 days. Majority or half of them were recovered and 2 of them were recovering. Unfortunately, now this is the important thing which I would like to mention here. Unfortunately, there was no information in the ICSR provided that what was the concomitant drug, whether it was a single drug, Montelukast, or some other drugs were also patient was receiving. What was the indication? why this Montelukast was prescribed. There was, I think, only one ICSR which reported indication. We looked for information in the package insert. In US, no, tinnitus is not reported. What information is provided is in the package insert is otitis media. In UK and India package insert, it is not included. It is not mentioned in the literature. We could not find any case reports or case series. However, the statistical values were positive. They were favorable. Look at this IC value. It is positive. The PRR value is more than 2. Chi-square value more than 4. However, we found these ICSR, the case narrative was extremely poor. It was extremely difficult to make out what happened to the patient, when the reaction happened, and when the reaction happened, how it was managed. The information was missing. The information on the concomitant drugs was also missing. The indication for Montelukast, there was only one case of allergic rhinitis. All 9 cases, the indication why Montelukast was prescribed was missing. What I'm trying to convey here is that we did not consider this as a causal relationship or a signal. The point I want to stress here is good quality reports, supporting data, and the evidence are the precious elements for potential signal evaluation. If the ICSR, they do not provide these critical information, it is useless. And this is a big responsibility on all ADR monitoring centers who are submitting the ICSR to the National Coordinating Center. With this, let's try to find out what are the different recommendations, actions which the signal review panel can take at the end of deliberations and discussions. You can see that these are the letters, forwarding letters, which are written by the Indian Pharmacopoeia Commission. And the first and foremost thing is if any of the drug, suspected drug, indication, and the adverse reactions which are observed in the database, and if the SRP members, panel discussions, if they consider, yes, this is a safety alert, we need to follow up, we need to discuss, monitor this. So the drug safety alerts are generated and they are communicated to the healthcare professionals, patients, consumers, to closely monitor the possibility of these adverse reactions and report to the National Coordinating Center Pharmacovigilance Program of India. So that is one thing, drug safety alerts. And that is, based on some discussions which the members found that yes, this is an alert. We need to have more reports and we need to inform, communicate the stakeholders. Second is the regulatory authorities. There are certain reactions and they are considered to be the signal. They are communicated to the regulatory authorities. The recommendations are done to the regulatory authorities to take appropriate actions, such as inform the manufacturers to include in the prescribing information, upload on the official website of the manufacturer, update in the package inserts or even promotional material. And the third category is if the SRP committee meeting, the members, if they are not convinced at that point of time by qualitative and quantitative assessment, the third recommendation is done is for follow-up. Wait and watch, look for more reports which may be submitted eventually, and then you can take it in the subsequent meeting. We need to have more information to confirm it as a signal. Last but not the least, poor documentation, poor case narratives, where they do not establish the causal relationship, the signal is closed. It is no more discussed. So what are the recommendations which the PVPI has submitted so far to the regulatory body, that is the Central Drug Standard Control Organization under Government of India. You can see so far up till now, 137 drug safety alerts are generated, communicated to the healthcare professional and the patients, consumers, updating the package inserts, conveying to the regulatory body, that is 54, and 7 signals are the hypothesis for subsequent assessment is already generated. And this is the list of those 7 signals: cefixime, which I already gave you, acute generalized exanthemus pustulosis, even doxycycline, fixed drug eruptions, cephalosporin sporine class, that is fixed drug eruptions, which we recently discussed. And this is, I think, a marathon task, excellent task, which is done by our National Coordinating Center. And what is the impact of this whole exercise? We all have been working, all the ADR monitoring center, the national center, the SRP members. So what is the impact? You can see that the whatever recommendations goes from the SRP, Signal Review Panel members, they are forwarded to the regulatory authority, and the regulatory authority, you can see the letters are issued from Government of India, Director General of Health Services, CDSCO, to all the state and union territory drug controller, which they submitted. That the letter goes to all the drug manufacturer, importers to revise the prescribing information leaflet. And you can see that even the pharma industry, the manufacturer, you can see the previous PIL, which was marketed for lamotrigine, the package insert has been revised, and whatever the signal which is identified by PVPI, it is now incorporated in the package insert. Similarly, the updated PIL of the marketed the pharma industry, you can see the Stevenson-Johnson syndrome, toxic epidermonecrolysis. These are serious type of skin reactions. The mortality is very high if the condition is not diagnosed and the drug is not stopped immediately because of the one of the fluoroquinolone. You can see that has been inserted in the package insert. And this, what I'm trying to convey here is whatever recommendations are goes from the National Coordinating Center, PVPI, to the regulatory body, and from regulatory body, it goes to the manufacturer. They are doing the job. And this is, I think, something excellent, which is, I think, worth appreciating. Moreover, the assessment, the identification of certain safety alerts, drug safety alerts, as well as the signals, they are also being shared globally by UMC and the WHO newsletters. You can see this is a very recent one. The tramadol-induced risk of urinary retention appeared in the WHO newsletter, SJ syndrome due to chloroquine, ofloxacin causing SJ syndrome, cefepime causing urticaria, cefotaxime causing angioneurotic edema, and again mefenamic acid and doxycycline causing fixed drug eruptions. And they are, what I'm trying to convey here is they are shared globally by UMC as well as by WHO newsletter. Hence, to conclude, signal detection is a stepwise process and is essentially a working hypothesis that indicates early drug safety problems. Secondly, the disproportionality analysis by VigiFlow, applying it to the pharmacovigilance database, it complements and adds value to the qualitative assessment. However, cannot replace or overtake the importance of medical review and judgment of pharmacovigilance professionals for final adjudication of causality. What I'm trying to convey here is that yes, there is still the importance of clinical assessment and clinical review. The statistical tools are complementary and they add value and they speed up to identify the drug safety issue. The contribution and the impact of the National Pharmacovigilance Database, National Coordinating Center, PVPI, has been significant and very positive. The drug safety alerts and achievements are regularly shared globally by UMC as well as WHO. Thank you very much.
Thank you, ma'am. Thanks a lot for your excellent presentation and narrating the steps for identifying signal detection. And you have also highly elaborated about the tools VigiFlow and VigiLyze for optimum use of signal detection. I'm sure the member states will be beneficial. I think now we'll Nikita, can we move on to panel discussion? What is next?
Yes, sir.
So I invite Dr. Meera Desai, Dr. Adrian, and Dr. Noha from WHO to kindly join with us for a panel discussion. If you have some questions and something to be clarified, all the experts are available with us, and also UMC experts are also with us. Dr. Jennifer and Dr. Tommy also requested to be there in the panel discussion. Dr. Jennifer and Tommy.
Thank you.
Yeah. So any questions, any kind of questions or anything to be asked? All panelists are with us. Dr. Jennifer is with us. Tommy is with us. Dr. Meera, Dr. Adrian, and Dr. Noha also from WHO headquarters with us. So we can have any questions and anything to be clarified.
Dr. Clive Salvan, if I can maybe start, I have a question.
Yeah, please.
Although I'm a panel member. So I had two questions for you, Dr. Meera. First of all, thank you so much for your presentation. It was really comprehensive and very well organized and very clear. I really enjoyed it. I had two questions. The first one was just out of curiosity. So you mentioned that the results of all this work and all this assessment that you do is then shared with the regulatory authority. But so far in the example that you saw, that you have shown, I've mostly seen some requests to label some adverse drug reactions. So I was wondering also if sometimes your recommendation were maybe larger, for example, with particular wording to include in, I don't know, the warning section or something like that. So for example, I don't know, with urinary retention, whether you would also recommend to include particular wording in leaflet on signs that should alert or something like that. Or if not directly, if you were associated in the future work of CDSCO to do that. So that's my first question. And the second question was just a little bit provocative because there is now that VG-based app. I was wondering because so we all agree that it's very important to have good quality cases, and I was just wondering how mobile app may influence the quantity of information that you would enter, because it can already be quite time consuming, to enter a lot of information, find the reports and so on a computer based. So I was wondering whether we were expecting any sort of adverse consequences of using apps, or whether at least we would want these preliminary alerts then to be completed in the future by more information. So these were my two questions. Thank you very much.
Okay, as I understand, the first question is that whatever recommendations from the signal review panel that goes from the National Coordinating Center goes to the regulatory authority, and the package inserts are revised. See, in between, there is one process. The recommendations which goes from the IPC, that is Pharmacovigilance Program, to the Central Drug Standard Organization, they also have a subject expert committee. So they place these all report recommendations depends upon the area. If it is related to dermatologist or if it is related to a cardiologist, they have a subject expert committee, and that is placed in front of the subject expert committee. The subject expert committee also deliberates on this and comes out with a recommendation. And that goes, that is approved by CDSCO finally, and then it goes to the manufacturer that yes, there is a need to revise the package insert. So I think I have answered your question. Right. Now, subsequently, suppose if, because the signal will not establish a causal relationship, you need to do further research. You need to undertake further studies. Now here, I think it is now the responsibility of the, I would say, other pharmacovigilance professionals to take up as a pharmacoepidemiological studies or certain other organizations. This will not be done, obviously. I think, Dr. Kalai Selvan, I am clear that this will not be done by the regulatory authorities to undertake further studies. That will be done, that can be done by some other academic organization, medical colleges, to take up these pharmacoepidemiological studies, the professional organizations, which are specially of that subject area expert, if it is a dermatologist, cardiologist, or rheumatologist, so they can take up and then finally confirm that this type of association is definitely confirmed. So I think I am able to answer your first question. You are mute, sir. Sir, you are mute.
Yeah, I could not unmute myself, sorry. But yes, you have answered your my question. Thank you very much.
And the second one, I think, what was the second question? Sorry.
So we're now moving towards the possibility to enter adverse drug reactions through mobile app. I was wondering whether you would expect that it may change somehow the quantity of information that is entered or the quality of the information there, and basically how to find ways to work around such possibility.
Well, I think as of now, it seems, because in India, almost 90% of the people have smartphones. So I think these type of apps, everybody is using. And we expect the number of reports will increase. However, at present, maybe Dr. Kalai Selvan can answer, what are the number of reports which are submitted through mobile app and the number of reports actually submitted by VigiFlow. So right now, I have no idea how many number of reports are coming from mobile app. But as of now, it looks to be very ambitious because people are using various types of app. And I think tomorrow we also have a discussion on increasing consumer reporting. So I think this can be very helpful to the consumers, provided it is available in the local vernacular language. The reporting form is very short and simple, or if it is generating a QR code. Nowadays, everybody is very useful to use this QR code, quick access code. So if there is a QR code, and if the consumers or a reporter, he may be a patient, he may be any other healthcare professional, a QR code is generated, which can be used in this mobile app. I think the signals will go to the nearby ADR monitoring center, and that's how the number of reports and the complete details can be included. So I think we are hopeful. It looks to be very exciting as of now, but I think time will tell.
Thank you, Dr. Meera. We have Dr. Adrian. Hope it is answered. Hope the things are answered. Thank you. Thanks a lot. So we have one more question to, I think, this UMC colleagues, maybe Dr. Jennifer or Dr. Tommy can address. Can the Vigi-based data access be provided to individuals such as from an ADR monitoring center or for research studies?
Thank you for your question, Dr. Gupta. The National Center decides who has VigiLyze access to the World Database, which is VigiBase. If you would like to see by a regional basis, the VigiAccess.org is available for the public. So if you enter a drug and a reaction, you'll be able to see in the region how many of those cases are available. But we also have, for research, you can also contact UMC for custom searches if you are given access by the National Center in India to have access to VigiLyze.
Thank you. Thanks a lot. I think Dr. R.N. Gupta is raising his hands. Dr. Gupta, you can add to whom you want to ask questions. I think one more question for related to. I think Dr. Tommy raised some question in response to SDG or similar to drugs of similar therapeutic class. They can be used usually the anatomic therapeutic chemical classification covers this, and some SDGs are based on ATC. That means that a certain, he answered the question already, has given the reply also. You can go through the chat box and you can read the reply as well. Madam Meera Desai, madam, you know Dr. R.N. Gupta is asking some of the questions to you. Can you check the chat box?
Okay, Dr. R.N. Gupta, what he is writing is that mefaminic acid side effects aplastic anemia and not below 12 years, but usually pedestrian. Pedestrian or pediatrician? Pediatrician prescribed syrup available in the market. So I think this has nothing to do with the signal review.
Yes.
And this is, I can understand there are so many drugs which they are not supposed to prescribe, and the clinicians, they prescribe. This comes under the different domain. Let me share with you here one, as per my experience, because I have been associated with pharmacovigilance before even PVPI was there, somewhere around way back in 2000, so almost 20 years. And you must be knowing as a pharmacology professor that there is one drug, nimesulide. And this nimesulide, way back in early 90s, it became very controversial because this was the drug which was available in the market, and there were reports from the country of its origin that this drug should not be prescribed to the pediatric patients. Now, today we have a good database. And I think especially in the tertiary care center, government hospitals, doctors are submitting ADR reports. And I had an opportunity to interact with the regulators the question which was given, that why this drug is available in the market, which is not to be prescribed for pediatric patients. The regulators, the answer was given is, we need to have some evidence. We need to have some proof based on that. Now, as you mentioned, there is not a single pediatrician who is reporting this, that this is the drug causing this reaction. How can the regulators, they take any action without any evidence? You know, India is one of the largest democratic country. If any action which is taken by the government without any evidence, what is going to happen? The manufacturers will come, they go to the court. And there lies the importance of ADR reporting. It is the responsibility of the prescribers to report it. Unless we have a substantial evidence data, then only we can convey it. This is the drug causing XYZ reaction, and that is why it should not be available for a particular group of patients. It should not be prescribed. And withdrawing the drug from the market, it is a very extreme step. That is the last thing. Only, yes, whatever action is taken by the regulators, they need an evidence. And I think it is responsibility of all the healthcare professionals to report. Unless they report, I think, again, we'll be back to zero. We know that it should not be prescribed, but there is no reports, no adverse reaction report. Who is going to take action?
Yeah, thank you, ma'am. Thanks a lot. Dr. Deepa, you have some questions.
Yes, sir.
Over to Dr. Deepalata.
A very good afternoon to one and all. At the outset, I would like to congratulate Indian Pharmacopoeia Commission, Dr. Vigilanandan, for his excellence and enormous efforts in organizing sessions on VigiFlow and VigiLyze, as well as I extend my thanks to Dr. Jai Prakash, sir, for conducting this program as a part of National Pharmacovigilance Week. Sir, this is an eye-opening to all the AMC centers. As a coordinator of Adverse Drug Reaction Monitoring Center from Hyderabad, I really thank you for organizing, as all the 3 sessions which have been taken are eye-opening to ADR monitoring centers. These are very useful. They will allocate and help us to report more in detail, especially the new concept of VigiMobile and entire detail about VigiLyze and Meera Desai ma'am. It was something like in detail description. So thank you very much for your sessions. And I have a small question, sir. May I?
Yeah, please. To
Yes.
whom?
Yes, sir. This is for Jennifer, ma'am.
Okay.
Hello.
Yeah, you can proceed.
Yeah. Yes, ma'am. You have talked about VigiMobile, right? We were thinking when we go to the field as an ADR monitoring center, we do keep going to patients and we do go for seeing how the reporting is going on in the peripheral sectors. Do you think this VigiMobile is accessible for the patients, ma'am?
Presently, it's been developed for the monkeypox situation that occurred. So it has the 25 core variables in the built into VigiMobile presently, and it is mostly geared towards the vaccine centers or the healthcare professional. So we would have to take that into investigation if that's something that would be something that the regional centers and the national centers would like to have. So we always listen to the people that are actually using the apps and that would like to have some different features. So that will be brought up. Thank you.
Thank you, ma'am. Thank you very much.
Thanks, Dr. Deepa.
Yes, sir.
Can we hear from Dr. Noga? Noga comments.
Sir,
can you unmute Dr. Noga? Nova, we want to listen from Dr. Noga from WHO headquarters. Vipin, can you unmute her?
Yeah, I think I've been unmuted. Thank you much, and thank you everyone for this very nice lineup of presentations. We went from using collecting data using different apps, which for me I think is a must because we are going into a digital world. So it's not just to increase reporting, but to keep up with reporting because people are moving from paper to digital. It's how we're moving. So it's a must. And we're very pleased to see that VigiMobile has been launched. And I know WHO, my colleagues from WHO have been working very closely with a UMC to produce this app. And we have a lot of hope for the future with this, that it would grow into something much bigger than just for AFIs, but then to be expanded to ADRs. And as someone just mentioned now, being able for patients to be used. And then moving on to the VigiLyze. VigiLyze, I mean, is a tool that has from the last, 5 years, I can say, or 6 years, has improved tremendously in the functions that it can do. I mean, I remember first using VigiLyze probably 7 years ago, and now it has come a very, very long way. It's improving continuously, and lots of new features are being added to make it much easier for countries to do signal detection in their home setting, and so it's a great tool, and we welcome these new developments with SDG and any further improvements that have come. And Dr. Mira, thank you very much for your presentation. It's very good to see how thorough India IPC is working with the signal detection. You brought up a lot of issues. You've told us, when you have such a big database, what you do, and you've highlighted that yes, you have these tools in places to have statistical signals, but this is not enough. You need to, it's just a tool to filter out when you have so much data, to filter out and to ask you what to focus on. But then you still need the technique of having to be able to look into the data and make conclusions. And you've made a very good point about the data and the quality. And hopefully, I think there was a point raised before about I think by Adrian, about whether the app would improve the quality. I don't know whether using the app will be maybe easier to contact the person reporting to get missing information. I don't know if that is the case. But there are ways that we do monitor the completeness of reports, and so maybe it'd be something to look into to see how complete reports are when it comes to using mobile apps. So I think it's I hope that countries have benefited from this, and for me it was very nice to listen in. And congratulations to you all.
Thank you. Thank you.
Thanks,
Dr.
Dr.
Kalaiselvan, can I ask?
Yes, ma'am.
Can I ask one question?
Yeah, please.
Yeah, my question is to Jennifer. This mobile app seems to be very exciting and something new. Now, is it already implemented in some countries? What is the experience?
Yes, we've had some pilot countries that have started to use it. So it's important to contact UMC if you are interested in using the app, so that you're on the list of countries that would be next. So.
Okay. But the response, because see, the ground realities of India is very different than rest of the countries. And we need to do some kind of even a pilot study here.
Yes, you're right, because having over 500 AMCs and the volume, that would be a large undertaking. So if that's something that India is interested, you can contact us.
Yes,
we'll
sure. Thank you.
With you.
Thank you.
So if no more questions, then once again, on behalf of Indian Pharmacopoeia Commission, the WHO.
Can I ask one question to Jennifer, ma'am?
Yeah, Nikita, this might be the last question. Yeah.
Yes, sir. Yes, sir. Is there any scope in future for collecting medical devices adverse event in VigiMobile?
Yeah, I can say no to the medical device at the moment. That's not in our scope.
Okay.
We are considering, like with the mobile app at the moment, it is made for immunization reports, and that the next step would be the adverse drug reaction reports, but medical devices is not in UMC scope.
Jennifer, is there any specific tools that WHO UMC is designing for medical devices?
No, we're not. I can say that.
Okay, thank you.
Thank you. Thanks a lot. So again, on behalf of Indian Pharmacopoeia Commission, WHO Collaborating Center for Pharmacovigilance, I express my sincere thanks to all the resource person, panelists, and delegates, particularly Dr. Adrian, Dr. Nohaj, Dr. Jennifer, Dr. Tommy, and Dr. Meera Desai, and all other experts and resource persons for making this event grand success. Thanks a lot for sparing your valuable time and inputs. I also take this opportunity to thank all the delegates who have joined this webinar. Thanks a lot to one and all. I wish you all the best. Thank you.
Thank you.
Thank you so much.
Thank you.
Thank you.
Thank you.
Thank you. Goodbye.
That is